Chapter 14, lecture capture, lesson number 3. Right. Let's first show this movie again.
The mitochondrion is the site of most of the cell's energy production. After food molecules are processed in the cytosol, they enter the mitochondrion, where they are further broken down. In the citric acid cycle, the molecules are stripped of high-energy electrons, which are donated to carrier molecules, such as NADH. The carrier molecules transfer the high-energy electrons to a chain of proteins called the electron transport chain, which is embedded in the inner mitochondrial membrane. The chain acts as a pump, using the energy of the electrons to move protons from one side of the membrane to the other. The pumping creates a proton gradient across the membrane, which the mitochondrion can tap to make the fuel molecule ATP. The electron transfer begins a multiprotein complex called the NADH dehydrogenase complex. This complex has a higher affinity for electrons than NADH and easily strips away the high-energy electrons. As the electrons are transferred from one protein to another in the complex, energy is released and used to pump protons across the membrane. Electrons are then transferred to ubiquinone, a different carrier that shuttles them to the next way station, called the cytochrome BC1 complex, which again pumps protons as they flow through it. Because each complex in the chain has a higher affinity for the electrons than the previous one, the electrons keep moving through the chain unidirectionally. Finally, cytochrome C delivers the electrons to the cytochrome oxidase complex, a third proton pump. The cycle repeats until the cytochrome oxidase complex has accumulated 4 electrons. From there, they are handed over to molecular oxygen. Oxygen takes up the electrons as it combines with protons, forming water as a product, thereby completing the stepwise path of the combustion of the food molecules. The mitochondrion is...
Okay, so let's start off by this is a very important part of the chapter. Let's start off by reviewing the major components of the electron transport chain. So there are 5 major components, and they are going to be the 3 pumping components and the 2 carrier components. So, of course, where is this thing going to be located? This is going to be located in the inner membrane of the mitochondria, right? The inner membrane of the mitochondria. And the 3 pumping complexes have a particular order, all right? So the 3 pumping complexes will have a particular order. And we call the first one the NADH, dehydrogenase complex. And the second one is the cytochrome BC1 complex. And then the last pumping complex is called the cytochrome oxidase complex. And again, these 3 complexes have in this particular order because we find that electron movement goes from left to right in the picture, and electron affinity of the members also go from left to right in the picture. Okay. All right, so these are the 3 pumping complexes, and what are they going to do? They're going to be responsible for pumping protons from the matrix side to the intermembrane space. All right, so we're going to be pumping protons out of the matrix into the area known as the intermembrane space, and we're going to be doing that at all 3 pumping complexes. Okay, so these are the pumping complexes, and we would really say the pumping complexes are really the guys that are doing the work because this is the major work of the electron transport chain. Major work of the electron transport chain is to do what? Is to pump protons and form a proton gradient. So therefore, the protons are going to be leaving the matrix, going across the inner membrane, right? Transporting across the inner membrane into the intermembrane space, right? And we would say that the electrons don't cross the membrane. They don't go from the matrix side to the intermembrane space. The electrons are basically moving through the complex, okay, moving through the membrane, okay, not across the membrane. So the electrons never cross from one side to the other. They actually are moving through the inner membrane from left to right. Okay, so they never cross the membrane. So make sure electrons don't move across. Electrons are not being pumped. Electrons are voluntarily moving. They're moving from left to right, as we just said. And this movement of their electrons from the left to the right is all exergonic. All exergonic. Okay, and of course, in order to move from one pumping complex to the next, we need some carrier molecules. And so we have a carrier molecule that's in between the first pumping complex and the second pumping complex, known as ubiquinone, otherwise sometimes called coenzyme Q. And then we have another mobile carrier known as cytochrome C that will take electrons one by one from cytochrome B1 complex to the cytochrome oxidase complex. Okay? So if we had to talk about these things, we'd say there's two carrier proteins, and we have to know where they're located. So the coenzyme Q, otherwise known as ubiquinone, is found between the first and the second pumping complex, and cytochrome C is a mobile carrier from the second complex to the third complex. And all of these things are considered to be protein complexes, because as we know, protein complexes are the most dynamic workers in the cell, except for one thing in this picture. Out of the 5 major components, one of them is not even a protein. One of them is a what? Is a lipid, and that's coenzyme Q. And coenzyme Q is a lipid that can carry up to 2 electrons at the same time, okay, from the first complex to the second. And cytochrome C is a carrier protein or mobile carrier that can only carry electrons 1 at a time. But this guy is a protein, right? So this one, all of them are proteins except for coenzyme Q, otherwise known as ubiquinone. Okay. And if we said, well, okay, where do the electrons come from? Okay, where do the electrons come from? The electrons have to come from the matrix side. And we know that NADH can donate a pair of electrons, okay, a pair of electrons to NADH dehydrogenase complex. And where does the NADH come from? The NADH would be coming from oxidative pathways such as what? Krebs cycle? And other ones too, like beta oxidation of fatty acids. Okay? And even can be theoretically coming from the high energy electrons that are generated during glycolysis via the 2 shuttles, right? Via the shuttles. All right, so the NADHs that provide the high energy electrons to the electron transport chain come in on the matrix side, and they come in, if they come from NADH, they come in, okay, from NADH going into the very first pumping complex. On the other hand, FADH2 can also donate a pair of electrons, right? Pair of electrons. But the high energy electrons from FADH2 end up coming into the components of the electron transport chain at the point of coenzyme Q, which is a sad problem. It's a little sad for for this guy, because by coming in at coenzyme Q, you miss the first pumping complex. Okay, and this is sometimes why we say that the electrons, even though the high energy electrons of NADH and FADH2 are really of the same energy level, high energy electrons, but because the electrons from FADH2 come in after the first pumping complex, and therefore is not as powerful as forming the proton gradient as NADH, okay, because you're going to miss pumping some protons as a result of coming in after the first pumping complex. We say in the long run, the electrons from FADH2 are less ATP efficient, So surprisingly, even though they possess the same amount of high energy, NADH electrons and FADH2 electrons, they basically have the same amount of high energy. But when it comes to transferring that energy and making ATPs, we find that the FADH2s are a little bit less ATP efficient. And this is because of where they have to come in. They don't come in. They can't come in over here. Okay, they can't come in over there. They only have to come in at the second complex. And of course, where does the FADH2s come from? The FADH2s can come from Krebs cycle and can also come from beta oxidation of fatty acids, and theoretically also come from which shuttle? I guess if I said which shuttle this one is, this one would be the malate shuttle. Okay, malate shuttle to come in as NADH. And then the other one would have to be what? The DHAP shuttle. DHAP shuttle. If you use the DHAP shuttle, your high energy electrons from glycolysis come into the electron transport chain as FADH2 electrons. But if you use the malate shuttle, okay, your high energy electrons from glycolysis will actually come in as NADH electrons. All right. And of course, the electrons, again, do not cross the inner membrane. They basically are running in the middle of the membrane. They're running across the membrane, but not being transferred or pumped across. The only thing that's being pumped across the membrane are going to be these what? Protons, correct? And then when the electrons finally get to the end of the electron transport chain, cytochrome oxidase complex has the ability now to give the electrons to molecular oxygen. Okay, so molecular oxygen is going to be the final electron acceptor, okay, in the electron transport chain. All right, so cytochrome oxidase complex has the ability to hold on to, have a binding site for molecular oxygen, O2. And then what will happen, as is said just in the little video, is that we have to accumulate, molecular oxygen has to accumulate a total of 4 electrons before cytochrome oxidase complex releases that reduced oxygen. So remember, the electrons are reducing molecular oxygen. And what do they reduce molecular oxygen into? You're reducing molecular oxygen because you're giving it electrons. Remember, Leo says Gur, right? If you gain electrons, you've been reduced. And what does oxygen gas get reduced into when it finally picks up what? 4 electrons? It gets reduced into, and let's take a look what would happen there. We would end up having, there would be 4 protons that would neutralize the 4 electrons here. And we'd end up having a silly formula that would be something like H4. Okay, O2, and H4O2 is a silly formula. That doesn't seem to represent any kind of stable formula, but H4O2 would actually redistribute itself as to 2 molecules of H2O. And that's why we say each atom of molecular oxygen, picks up a pair of electrons and gets reduced into nice, safe, happy water, H2O. Okay. All right, so just reviewing a little bit, and you have to know not only the order, okay, but you have to know like what are the components, who are the mobile carriers, are all of them proteins, is one of them a lipid, which component would have a higher electron affinity, And electron affinity is considered to be the attractiveness, okay, of something to an electron. Does electron want to go? If something has high electron affinity, that means electrons want to go there. And that if given the opportunity, electrons will spontaneously move from an area of low electron affinity, if given the opportunity, to an area of higher electron affinity. This is why the movement of the electrons from left to right in the membrane, they don't go from one side of the membrane to the other. They just move through the middle of the membrane from left to right. Okay, that they do this exergonically because of the way the electron transport chain is set up. That when the electrons get the opportunity to see the next component, they go, oh, the next component has higher electron affinity. I want to go there. And the electrons spontaneously, exergonically move from one component to the next, moving from left to right without any expenditure of energy. We're not pumping electrons, we're pumping protons. Students always get this confused. They think it's called electron transport chain. They think that they're pumping electrons. No, no, no. Electrons are not being pumped at all. Electrons are spontaneously moving through the electron transport chain in the hopes that they will finally see something with very high electron affinity. And something that has very high electron affinity is the element oxygen, as we've talked about before in the previous chapters. So it's the promise of eventually keep moving forward from left to right, and you will see oxygen. You will get to go to oxygen, and the electrons are like, wow, that's what I want to do. Now, of course, what happens if cytochrome oxidase? What happens if cytochrome oxidase prematurely releases the oxygen before it's fully reduced? And again, this is one of the risk factors of the electron transport chain, to be able to hold on to molecular oxygen and try to hold on to it tight enough so that all 4 electrons have an opportunity to get there, is sometimes a challenge, especially for an old electron transport chain that may not be functioning as well. And even if it wasn't an old one, there's always going to be the opportunity for mistakes where molecular oxygen will leave the electron transport chain or escape from the electron transport chain without being fully reduced into water. And so therefore, if we had molecular oxygen that only picked up 1 electron and then escaped, then what we do is that we say we have the what? We have the oxygen-free radical. We have the oxygen-free radical, okay, the superoxide-free radical, which is a bad thing, right? The superoxide-free radical can cause oxidative damage and oxidative stress. So this is bad. This is bad. And what happens if molecular oxygen only ends up picking up 2 electrons? So if molecular oxygen only picks up 2 electrons, then we would have O2 with 2 electrons. It will no longer be a free radical. It won't be a free radical like the superoxide free radical, okay? But it will still be bad because it will get reduced. It will get neutralized with 2 hydrogen protons and become H2O2, hydrogen peroxide. And what happens if the oxygen picks up 3 electrons? So we're getting so close now, we don't have 4, okay? Then what it turns into is that it will turn into, okay, this will be neutralized by 2 protons, water, but the other oxygen out of the O2 would end up picking up only 1, will turn into the hydroxyl free radical, which is really bad. So this is a big problem with using oxygen in aerobic respiration, because at the end, why we're using oxygen, we're using it as the final electron acceptor. And it's really dependent upon that electron transport chain to make sure that O2 gas gets fully oxidized, gets fully reduced with 4 electrons before it's allowed to leave the electron transport chain. And if oxygen gas gets incompletely reduced with 1, 2, or 3 electrons, It causes the formation of free radicals, such as the superoxide free radical, and the worst yet, the hydroxyl free radical, okay, which is the worst free radical, okay? And it also can produce or can be a source of the production of hydrogen peroxide, which also, while it's not a radical, causes oxidative stress. So, this is sometimes referred to as high-risk biochemistry, to actually play around, okay, with molecular oxygen and trying to get molecular oxygen to what? To pick up the electrons one at a time and have to stay there partially reduced until it's fully reduced with 4 electrons, because it is a source then of what? Oxidative stress, because of these radicals that can form at the electron transport chain and the hydrogen peroxide that can form. So that's a high-risk biochemistry, but luckily, most of the time, okay, things are working out what? Things are working out well, and oxygen gets fully reduced into 2 molecules of what? Of water, all right? But this is actually the number one source of oxygen radicals in your body is the electron transport chain, the number one source of oxygen radicals, like the superoxide free radical and the hydroxyl free radical, and probably the number one source even of hydrogen peroxide that's generated in your body is caused by the electron transport chain, okay? But again, we're happy with the electron transport chain. We're happy with what? Oxidative phosphorylation, because it makes tons of what? It helps in the process of making tons of ATP for the cell, way better than anaerobic respiration, right? So you kind of live a hard and fast and rich life at the same time. All right, so some of the energy released during electron transfer can be conserved, right? So we're talking about the electron transport chain, and we want to conserve some of the energy that's released during electron transfer. That electron transfer, each step of electron transfer is called a redox step, a redox step. And a redox step is an oxidation reduction step, okay? And the redox step generates energy, free energy. I should say free energy. Doing a redox step generates free energy. And remember, free energy, you either use it or you lose it, right? If you use your free energy, if you're smart enough, you could use it to do some kind of work. And if you don't use that free energy, or if you can't completely use that free energy to do work, you know what's going to happen to it. It's not going to be conserved to do some sort of work. It's going to be lost as what? As heat energy. So the energy that's released during electron transfer, which is during the redox step of the electrons moving from member to member in the electron transport chain, can be conserved. Not all of the energy can be conserved, but some of it can be conserved. Conserved to do what? To do some kind of work, right? And what kind of work does the electron transport chain really do? They do what? Proton pumping. The electron transport chain conserves some of the energy in the form of a proton gradient, because when you do proton pumping across the membrane, you form a proton gradient. So this free energy of the redox reactions of the electron transport chain, okay, is used to do work. And that work is pumping protons across the membrane to form a proton gradient. And that proton gradient represents a battery, stored energy, because isn't that what a battery is? It represents stored energy. So we conserve the energy that's released during the redox steps of the electron transport chain. We conserve that to do proton pumping to charge a battery, to form a proton gradient, which is really the same thing as a battery. It's stored energy across the inner membrane. Why didn't the early researchers realize what the real job was of the electron transport chain? Just like many students think that the electron transport chain changes ADP into ATP, that's what the early researchers thought too. They said, well, there must be some member of the electron transport chain that's using the energy to phosphorylate ADP to ATP. But when they were studying the components of the electron transport chain, trying to figure out what they could do, what were they doing with these redox steps? Okay, they were trying to figure out, well, what is the job of the electron transport chain? We know what goes in, we know what comes out. I don't understand how they are conserving the energy and making ATP. And they found out, sure enough, none of the members of the electron transport chain was able to change ADP into ATP. That's not its job. And because they were purifying the components, the protein components out of the membrane, and then trying to study, well, what are these guys doing? Who's the one that can change ADP into ATP? And they couldn't find any of them doing that when they were purifying them out of the membrane. And so therefore, for a very long time, they couldn't figure out what the job of the members of the electron transport chain were. They didn't know what the jobs were. And that's because they were taking them out of the membrane. Because as soon as you take them out of the membrane, even if you were pumping protons, you can't form a proton gradient because you don't have a membrane barrier anymore, okay, to form the proton gradient. All right, so they didn't realize what the real job of the electron transport chain was, which was to pump protons and form a proton gradient across the inner membrane, because they were purifying these proteins in the membrane. Go, what do you guys do? We don't understand your job and your function. All right, so it really wasn't until people said, well, let's see, maybe the membrane, an intact membrane, is important, okay, to determine what the job of the members of the electron transport chain really were. All right, what would happen to the energy released during electron transfers if no protons were pumped? Okay, suppose that the pumping complexes, which are really like machines that know how to use the free energy of the redox reaction to conserve it like a machine and do some sort of machine work, right? Suppose that these pumping complexes didn't pump protons, then what would happen to all that free energy that would be released with the movement of the electrons? It would all be lost as what? It would all be lost as heat, correct? Because remember, okay, anytime you release energy, first you release it as free energy. And then that free energy can be used to do some kind of work. If you have a machine, you have to have some sort of machine that knows how to utilize the free energy to do work. And remember, you can never use all the free energy to do work, because who says you're not allowed to do that? The queen of thermodynamics. But the electron transport chain is considered to be, work-wise, about 50% efficient, pretty much maybe approaching the limit that the laws of thermodynamics would actually allow. That's how beautiful the energy, the free energy that can be released from these redox reactions have been captured, that the overall effectiveness of just the electron transport chain when it comes to utilizing the free energy to do work is close to 50%.